Hey everyone, and welcome to this explainer. Today we're doing something truly fascinating. We are going to decode the incredibly precise, almost clockwork-like chemical orchestra of the female reproductive system. By using a comprehensive clinical text as our guide, we're going to strip away all the confusing jargon and look directly at the brilliant biology that drives this system, starting from before a female is even born all the way through adulthood.
Okay, let's dive right into our roadmap for today. We'll be following a chronological timeline. Starting with 1, the oocyte countdown. Then we'll move into 2, puberty's awakening. 3, the hormonal orchestra. 4, the menstrual cycle. And finally, 5, when timelines shift.
Starting off with Section 1, the oocyte countdown. So, when do you think a human female actually has the absolute highest number of eggs, or oocytes, in her ovaries? You'd probably guess puberty, right? Or maybe right at birth. Well, the truth is actually wildly counterintuitive. It's 6 to 7 million. That is the peak germ cell count. But here is the absolute kicker. This peak occurs at just 20 weeks gestation. Think about that. A female reaches her absolute maximum potential for fertility when she is merely a 5-month-old fetus inside her mother's womb. Before she even takes her very first breath, her biological clock is already ticking. And this brilliantly illustrates what happens next. From that massive peak, it's basically a story of rapid, continuous decline. From 6 to 7 million at 20 weeks gestation, the number just plummets to about 1 to 2 million by birth. By the time she hits puberty, around age 12, there are only about 300,000 to 400,000 left. And by menopause, at around age 50, fewer than 1,000 remain. You see, the human ovary has a fixed pool at birth, and it only depletes through a process of natural cellular decay called progressive atresia. Unlike your skin or your blood, which are constantly regenerating themselves, this oocyte pool is strictly non-renewable. There are no stem cells making new eggs later in life. What you are born with is literally all you get. And honestly, even wilder, these remaining primordial follicles are essentially frozen in time. They are arrested in a phase of cell division called prophase 1. They just sit there, perfectly still, waiting for years, sometimes decades, until right before ovulation to finally complete their development. Moving along to Section 2, puberty's awakening. After years of childhood quiescence, where the reproductive system is basically just asleep, a whole cascade of hormonal events jolts the system awake. And the sequence of puberty generally follows a very specific order. It usually starts with a tharlarch, which is breast development, around age 10. That's followed by adrenarche and pybarch, the development of pubic hair, which is actually driven by DHEA from the adrenal glands. Next up is peak height velocity, around 11 and a half years old. And finally, menarche, the very first menstrual cycle at a mean age of 12.5 in the US. Interestingly enough, prolonged exposure to estrogen is what eventually causes the growth plates in the bones to fuse, limiting adult heights. And fun fact about those early cycles right after menarche, they're often anovulatory. Basically, the body's practicing the hormonal cycle but not actually releasing an egg yet. But what actually flips the switch to start this whole process? Well, it comes down to genetics, specifically an epigenetic switch. During childhood, you have this paternally expressed gene called MKRN3, and it acts like a really heavy foot on a brake pedal, just suppressing puberty. When the time is right, that brake is released, and that allows a network of kisspeptin neurons in the brain to activate and kick off the pubertal cascade. Which brings us to Section 3, the hormonal orchestra. You can really think of the female reproductive system as a profoundly complex symphony. The brain acts as the conductor, and the ovaries are the instruments playing the music. Our conductor here is a molecule called GnRH, or gonadotropin-releasing hormone. Now, what's really interesting about this is where these GnRH neurons come from. During fetal development, they actually originate in the olfactory placode, the area responsible for your sense of smell. And they literally migrate through the skull along the olfactory nerves to eventually settle in the hypothalamus in the brain. It's pretty wild. And from there, they pulse rhythmically to direct the entire reproductive cycle. Here is how that chain of command works down the hypothalamic-pituitary-ovarian axis. First, the hypothalamus pulses GnRH, and the speed of these pulses, absolutely crucial. Slow pulses tell the pituitary gland to release FSH, while fast pulses tell it to release LH. Finally, FSH and LH travel all the way down to the ovaries, prompting them to grow follicles and produce our big players, estrogen and progesterone. Down in the ovary, producing that estrogen requires a microscopic assembly line called the two-cell model. It takes major teamwork. The theca intercells sit on the outside of the follicle. They grab the LH signal and manufacture androgens. Yup, male hormones. but they can't finish the job. So they pass these androgens to the mural granulosa cells inside the follicle. The granulosa cells receive the FSH signal, which turns on an enzyme called aromatase, and that converts those androgens directly into estradiol, the primary estrogen. It is a stunningly precise division of labor. So, what do these hormones actually do? Well, they create tangible physical changes in the body. Estrogen is essentially the builder. It thickens the uterine lining and creates this thin, watery cervical mucus that is super friendly to sperm. Progesterone, on the other hand, is the stabilizer. It matures the uterine lining in the secretory phase, it turns that cervical mucus into a thick barrier, and get this, it even raises a woman's basal body temperature by over 0.5 degrees Fahrenheit. But there's one more really crucial chemical we need to mention, AMH, or anti-Müllerian hormone. So the crucial point is that AMH acts as a biological gatekeeper. Remember those hundreds of thousands of resting eggs we talked about? Without AMH restraining them, they would all try to develop at once and just burn out immediately. Because AMH is produced by the small, growing follicles, it actually serves as the best, most stable clinical marker we have for a woman's remaining ovarian reserve.
All right, moving to section 4, the menstrual cycle.
This is where the hormonal orchestra puts on its magnificent, synchronized monthly show. A normal cycle is 25 to 35 days, broken down into distinct phases. It starts with the early follicular phase, where a rise in FSH recruits a new batch of follicles. By the mid-late follicular phase, a single dominant follicle is selected and it starts pumping out high levels of estradiol. Then comes ovulation, followed by the luteal phase, where the collapsed follicle, which is now called the corpus luteum, secretes progesterone. What is so fascinating here is that while the first half of the cycle can vary greatly in length, the luteal phase is strict. It is a fixed 12 to 14 days pretty much every time. So with all this microscopic biochemistry happening deep inside the body, it brings up a very practical question for women and clinicians alike. How do we actually know ovulation occurred? Well, the clinical text gives us several ways to track this. The absolute gold standard is a blood test showing serum progesterone over 3 nanograms per milliliter, usually checked about a week after presumed ovulation. But you can also track it right at home. At-home ovulation predictor kits, or OPKs, detect the surge of LH in the urine, which happens about 36 hours before the egg releases. And that basal body temperature rise we talked about earlier, tracking that every morning gives you fantastic retrospective confirmation that, yes, ovulation definitely happened. Finally, section 5, when timelines shift. Let's move to our last major concept and see how this builds. What happens when this intricate clockwork runs too fast or too slow? When the clock runs too fast, we see precocious or early puberty, and we divide this into 2 categories. Central precocious puberty is when the brain's axis activates too early. The conductor, GnRH, is driving the process, so doctors treat it with GnRH agonists to essentially hit pause on the brain's signals. Peripheral precocious puberty, however, is totally independent of the brain. It's caused by the autonomous production of estrogen, maybe from a cyst or a tumor. Because the brain isn't driving this one, treatment requires targeting the hormones directly, often using aromatase inhibitors. Conversely, when the clock runs too slow, we see delayed puberty, and this really highlights how deeply the reproductive axis is tied to both our genetics and our environment. For instance, Turner syndrome is a genetic condition where a missing X chromosome leads to early loss of oocytes and delayed puberty. But the system can also be stalled completely by environmental factors, like in functional hypothalamic amenorrhea. If a young woman is experiencing a severe energy deficit, say from extreme exercise or undereating, the brain literally senses that lack of calories and shuts off the GnRH pulses to conserve energy. It basically delays puberty entirely. As we wrap up this explainer, I want to leave you with this final thought. The female reproductive system is a non-renewable countdown, managed by a profoundly delicate chemical orchestra. It truly is a marvel of human biology. From millions of cells waiting in the wings before birth, to the rhythmic pulses of the brain guiding a monthly cycle, the precision and fragility of it all is just awe-inspiring. So it makes you wonder, what other microscopic symphonies are playing inside of us right now, just waiting to be understood? Thanks for learning with me today. Keep asking questions and keep exploring.